Welcome back. We are now in chapter 21, part C. And this is your cell mediated immune response. We have just finished the humoral immune response, the production of antibodies. Now we're going to get into the realm of the T cells, the killer T cells and the helper T cells and the regulator T cells. So T cells provide defense against intracellular antigens. Example, for instance, of these antigens will be your cells infected with viruses or bacteria, cancers or abnormal cells, foreign, such as transplanted cells. Some T cells directly kill. Others would not kill, but they will release chemicals that regulate the immune response. Cells that would directly kill will be what you call the killer T cells, and the regulator ones will be your regulator T cells and your helper T cells. So let's take a look at the major types of T cells. The first one is actually the CD4 cells. The CD4 cells, and let me show you what a CD4 cell is. This is your CD4 cell. And the CD4 cell usually become your helper T cell. So this one is a CD4. This one doesn't look like CD4. It has a different color and it's a different code. It is a CD8 cell. The CD4, I don't think you know what I'm pointing at. Now you will know. Okay, this is your CD4 cells. CD4 cells can help activate B cells and other T cells, as well as your macrophages and the direct adaptive immune response. They can regulate that because they are there to help. I tell you, without the helper T cells, guys, our immune response will collapse. One thing for your fact sheet, the AIDS virus. The AIDS virus will attack the helper T cells. And that's why the immune system of people with AIDS, once they have the full-blown AIDS, their immune system will collapse. And a lot of things can happen. They can get sick and nothing is going to help them. Nothing is going to protect them. It's because your helper T cells that used to be CD4 has been compromised by the AIDS virus. So some of the CD4 cells become regulatory T cells, which moderate immune response and can also become memory T cells. What are memory T cells? Memory T cells are the cells that has in their memory the ability to um, recognize an antigen. And therefore, if there's going to be another infection, a secondary infection of the same antigen, then they can already like spring back into action. They don't need to be primed anymore. Remember what I mentioned to you in in our previous discussion. Priming, the initial exposure will take priming, and priming takes time. The second time that we get the same antigen, then there won't be any more priming and the reaction will be immediate. The CD8, this is actually the CD8. The CD8 becomes the cytotoxic T cells, or otherwise known as the killer T cells. They are capable of destroying cells that are harboring the foreign antigen. They also become memory T cells. They are memory T cells of these antigens, of the antigens that they were exposed to. Those naive CD4, and CD8 cells, those T cells that has not been exposed to any antigen, you call them naive. You do not call them helper T cells or cytotoxic T cells. No, they are just CD4 and CD8. And they could be, in the lymph node waiting for their chance to meet the antigens or to be exposed to the antigen. But once they're exposed to the antigen and they get activated, you don't call them anymore CD4. They turn into a helper T cell. CD4s will turn into helper T cells, and the CD8s will turn into cytotoxic T cells. CD4s can also turn into suppressor T cells. So, your helper, cytotoxic, and regulatory T cells are already activated T cells. Naive T cells are called just plainly CD4 or CD8 cells, just like what I said. T cells respond only to processed fragments of antigen displayed on the surface of cells by major histocompatibility complex proteins. Antigen presentation is vital for activation of naive T cells and normal functioning of effector T cells, just like what I mentioned to you in the previous video. Our real heroes in the battlefield, one of the, to me, is actually the antigen presenter, because they could, in the process, after they have been after they have phagocytized, these are the phagocytes, after they have eaten and taken in the microorganisms and digest all the fragments, they're going to be showing them in the plasma membrane, and by showing them in the plasma membrane, they could be visualized by other cells, by other body cells, to be foreign, because they are carrying the foreign antigen, and therefore they can be attacked and they could be killed. But then again, they needed uh, a non-moving or maybe slow-moving white blood cell to be around, so that the other white blood cells will recognize them as, or will recognize the antigens that they are already carrying. So this thing mentioned something, this page here mentioned something about major histocompatibility complex. And there are two classes of major histocompatibility complex proteins. You call them MHC so that they will not be tongue twisters. There are two classes, the class 1 MHC and class 2 MHC. Class 1 is displayed by all cells except the red blood cells. Class 2 proteins are displayed by the antigen presenting cells, such as your dendritic cells, your macrophages, and your B cells. Both types are synthesized in the endoplasmic reticulum, and they bind to peptide fragments. Let's review this, because we've already taken this in the previous, not previous chapter, but previous part of this chapter. Um, these major histocompatibility complex are coded by our genes or in our genes, and they are unique to every individual. And this will be your MHC1. It is just a cartoon diagram, and this is your MHC2. MHC1 has transmembrane segment, a single transmembrane segment that is wedged right into the plasma membrane. And the MHC2 molecule has two transmembrane protein that is actually deep into the plasma membrane. And they both have peptide binding, protein or peptide binding cleft. This group pretty much is actually where your antigens can actually dock in. Take note, T lymphocytes can only recognize antigens that are presented on the MHC proteins. They will not be recognizing the T lymphocytes that we're talking about now, will not be able to recognize any antigens that are floating in the blood. They will not know that. They can only recognize the antigens if they are presented by the APC, the antigen presenting cells. So, needless to say, whether we need the APCs or not, yes, we need them very badly. So let's take a look at the first MHC protein, MHC1. They bind with short fragments, about 8 to 9 amino acids of endogenous antigen. The proteins synthesized these proteins are synthesized inside a cell, and you call them endogenous because they are synthesized inside the cell. And they could be self-antigen, normal proteins of the cell. They could also be non-self. These are abnormal proteins, but you don't find them just around. You find them in infected or abnormal cells. So you find them in a cell, but the cells should be infected or abnormal. That's the reason why you call them non-self antigen, because they don't look like they are the normal cell. So the MHC1 is crucial for CD8 cell activation, because this is actually this MHC. The MHC1 is the one that activates a CD8 into killer T cells. The class 2 MHC protein bind with longer fragments, about 14 to 17 amino acids of exogenous extracellular antigens that have been pretty much like engulfed or broken down by an antigen presenting cell. The class 2 proteins are recognized by helper T cells, and they signal the CD4 cells that help is actually required. And right here, again, just like any other chapters that I will be presenting to you, I will always be presenting to you tables of um, proteins or structures and their function and whatever importance they have and the information they have. This is going to be very important, and you can use it for your review. You can always, like in this video or um, in your textbook, in your ebook, you can always look for this and study it just like the way it is. Or look into my video and pause it in this particular page so you can study it. Major histocompatibility restriction. Both CD4 and CD8 cells have different requirements for MHC protein that presents antigen to them. First, your CD4 cells that become helper T cells are restricted to binding only class 2 major histocompatibility complex proteins, typically on the antigen presenting surfaces. The CD8 cells, on the other hand, become cytotoxic T cells and are restricted to binding only class 1 MHC proteins found on every cell surface, including the APC surface. Once activated, your cytotoxic T cells seek out the same antigen on class 1 MHC protein on any cell. The dilemma of APCs, or the antigen presenting cells, is how to enable the CD8 cells to be activated by non-endogenous phagocytized proteins. That is really going to be very hard to do, because your bloodstream is actually flowing and they are really fast, and all of the cells are passing through, and nobody will find something that needs to be recognized in a very powerful um, movement and a powerful flow and strong flow of blood. And that's going to be the dilemma. The dendritic cells, these are your dendritic cells. These are antigen presenters of the skin. They are Langerhans cells. They get around this problem by engulfing dying virus infected or tumor cells. And this is going to be the story that I mentioned to you already, how they become our heroes. They engulf them, and then they digest the antigen, and they display bits and pieces of the endogenous proteins on both the class 1 and class 2 MHCs. The dendritic cells can also import antigens via temporary gap junctions with infected cells rather than engulfing them. And that's really neat. T cells can be activated only when antigen is presented to them. Activation is a two-step process. The first step is antigen binding. So here, take this and take note of this. Your T cells will not become helper or killer T cells if they are not going to be activated in the two-step process. They cannot only be presented by the antigen and then voila, they already know what to do. No, it has to be a two-step process. Somebody will tell them, hey guys, come here, look at this. I'm presenting to you, this is your enemy. But that's all? No, that's not all. There is a second step, which is a co-stimulation. Now, a helper T cell or a killer T cell will need both antigen binding, which means they need to see the antigen and be able to touch the antigen. And they need to be co-stimulated by another protein. Both occur on the surface of the same APC. Both are required for the clonal selection of T cells, which means before they can clone themselves because they know already what to do, they're already real soldiers and they can produce, they can already fight. Before they can even do that, they have to go through these two process, the two-step process. The step one is antigen binding, and here you can see the antigen binding to the T cell or the CD48, or I don't know if this is CD48 or CD4 CD4 or CD8. Let's see. The T cell antigen receptors bind to the MHC complex on the APC surface. Your APC is right here, and the MHC is right there. The APC is actually handling or is actually presenting the antigen via the MHC. Remember, your lymphocyte T cannot know what you're giving to them, not unless you present it to them in an MHC. It's just like the MHC is a platter, a gold platter, and they're not going to be looking at your antigen if you are not going to present it to them in a gold platter. Interesting. All right, so the the there's actually supposed to be the presentation must be like a double recognition by recognizing both the MHC antigen and the foreign antigen. So the MHC needs to be recognized and the foreign antigen needs to be recognized. This means only one thing. If that was a self antigen, your T cell will say, go away. I'm not interested in your antigen because that is self. I am not supposed to touch the cell. So all of the T cells that come out of this plane, remember that they actually gain immunocompetency or the ability to become good soldiers right there in the thymus gland. The moment they get out of the thymus gland, they should know already what is self. If they don't know self, they cannot leave the gland. They're going to be destroyed right there. So, if there's actually double recognition, they know that it's actually an MHC presenting it. It's a double. It's actually a gold platter, and it is not self. Then it is actually going to study what the antigen is all about. Then the second part is a co-stimulation. Now, complete T cell activation requires T cell also to bind to one or more co-stimulatory signals on the surface of APC. Without co-stimulation, there is going to be what you call anergy. What is anergy? Well, anergy is uh, a state by which a T cell would not know what to do, even if they were presented with an antigen without the co-stimulation coming from this very special protein and chemical inside the APC, this soldier will not know what to do. It's just like calling on to a soldier and saying, hey, soldier, this is actually I'm presenting to you your enemy. And the soldier is looking at the enemy. The soldier knows that it is the enemy, but the soldier cannot do anything. Why? Simply because at that moment the soldier needs to get a co-stimulation or a prodding, probably from his boss, probably from the sergeant, saying, okay, soldier, kill. Of course, that doesn't happen in reality because a soldier is supposed to be given his own way of decision, but not your T cells. Your T cells would need a co-stimulation from interleukins and special proteins, because without those co-stimulation, then they are not going to be able to work. The third one, after they have already recognized what the antigen looks like, they're now going to start to proliferate and differentiate. Remember that if there's only one T cell and this one T cell is able to kill, that one T cell is not enough to kill all millions or hundreds and thousands of pathogens that are already spread out in a bloodstream. He's going to be killed by doing that. So what he needs to do first will be just like your white bloods, your lymphocytes. These T cells that has been stimulated, that has been activated, will need to clone itself into millions of T cells that would have the recognition of the antigen. T cells are activated, they enlarge, they proliferate, they clone themselves in response to cytokines. They differentiate and perform functions according to their T cell class. If they are helper T cells, they will need to help. If they are killer T cells, they will need to kill the microorganisms or cells that would carry that same antigen. And here is the clonal selection itself. You have here a class 2 MHC protein displaying a processed bacterial antigen, and you have a co-stimulatory molecule that is already there. Now, both after the presentation, the co-stimulatory molecule is already there, and therefore after the presentation, you can be sure that your T cell is already going to be good to go. So the first one is actually your antigen presentation. And right here, what you have here looks to be like, well, it looks like a dendritic cell. The dendritic cell is already presenting the antigen. Then in the second one, you have double recognition. The CD4 T cell is now recognizing the antigen MHC complex. It is not a self. It is actually a foreign antigen. Both the TCR and the CD4 protein bind to the antigen MHC complex. The co-stimulatory molecule is now going to stimulate or co-stimulate the CD4 into becoming either a regulatory cell or a helper T cell. After being co-stimulated, you now have the clones here. Well, that particular cell is now capable of cloning itself into CD4 T cells, and one of them will end up being a memory cell. The others will continue to work, and this time, these T cells here are actually helper T cells. So let's take a look at some molecules that help in the co-stimulation. One of the molecules is actually what you call the cytokines. These are chemical messengers of the immune system. They mediate the development, differentiation, and responses in the immune system. These cytokines would include interferons and interleukins. I think I must have already discussed with you what interferon is as with the reaction or with the role of the interferon in preventing viral spread. Okay, so interleukin 1 is released by macrophages and they stimulate the T cells to release interleukin 2 and they synthesize more interleukin 2 receptors. Interleukin 2 is actually a growth factor, and this growth factor is actually a co-stimulant, and they will be co-stimulating a CD48 and a CD4 and a CD8 into becoming a helper T cell or a cytotoxic T cell. And these are your interleukins, the interleukins and the function of the interleukins in their immune response. Now let's take a look at the helper T cells. The helper T cells help in the humoral immunity. Pretty much what is the central role of the helper T cell in mobilizing both the humoral and cellular immunity. Now, the helper T cells are pretty much like very important. As a matter of fact, the AIDS virus I mentioned to you, they actually immobilize the helper T cells. And when the helper T cells are immobilized, the entire immune response of the individual is gone. The helper T cells play a central role in adaptive immune response, and they activate both the humoral and the cellular arms. Once they are primed by the antigen presenters, the helper T cells could help activate B cells and other T cells. They induce T cells and B cell proliferation, and they secrete cytokines, and they recruit other immune cells. Without the helper T cells, there is no immune response. So, helper T cells become activated when they interact. Well, helper T cells interact directly with B cells while displaying the antigen fragments that are bound to the MHC2 receptors. They stimulate B cells, the helper T cells would stimulate B cells to divide more rapidly and begin antibody formation. This is actually in the humoral. The B cells may be activated without the helper T cells by binding to T cell independent antigens, but the response is weaker. It is not the same, um, when the signal and the binding and the activation is coming from a real helper T cell. That's actually now the response is going to be uh, long-lived and is stronger. Most antigens that are T cell dependent antigen require the helper T cell co-stimulation to activate the B cells. The CD8, the CD8, uh, the CD8 cells. Let me just give to you the CD8. There you go. This is actually a CD8. I have the CD8 protein. So we have here CD8 below, the helper T cell is right there on top. Whoa, boy, you have here an antigen presenter, a dendritic cell that can activate both helper T cells and a CD8 cell. The CD8 cell require a helper T cell to become activated into destructive killer T cells. They cause dendritic cells to express a co-stimulatory molecule required for CD8 cell activation. The role of a T cell in amplification of innate defenses pretty much is that they amplify innate immune system, they activate macrophages, leading to more potent killers. They mobilize lymphocytes and macrophages and attack other type of white blood cells around. Pretty much um, your killer T cell really is an active cell. When they are activated, they go out there, they clone themselves, and all of them are all ready to fight. There you go. Your dendritic cell has now activated your CD8 cell with the help of interleukin 2. And now your CD8 cell is now a killer T cell. So the killer T cell, just like what the name implies, will attack and kill other cells. The activated killer T cells will circulate in the bloodstream, in the lymphoid tissues. They go to the lymphoid tissues in search of body cells that would display the antigen that they can recognize. The activated T cells will target virus infected cells, cells with intracellular bacteria or parasites, and even cancer cells and foreign cells, as in the cells that are actually with recognizably foreign because they are transfused, they're foreign blood, or transplanted, they're foreign tissues. The cytotoxic T cells will pretty much deliver a lethal hit, causing two mechanisms. The first, and this is actually something like how they do it. I mean, if in my former students, the way I explain it to them will be like, for instance, there is actually a, a house where this soldier knows there's actually a lot of enemies there. What will the soldier do? Well, probably the soldier will think that I don't have any bullets with me, but I have grenades. So I'm going to open one window. So he goes there at the back, opens one window, and throws in a grenade, and the entire house will explode. That's neat, right? You can only see that in the movies. But that thing really happens in your cytotoxic T cells. The T cell releases perforins. Perforins are substances that create pores. It's just like opening a window of the cell, all right? And granzymes. Granzymes are enzymes and they can stimulate apoptosis, meaning bursting of cells. So imagine your your T cells, your killer T cells, creating a hole in the plasma membrane of the enemy of the bacteria, and then putting enzymes in it, and then the bacteria will explode. Boom. That's the way your killer T cells move. And but it is one on one. Remember that. It is not as effective as, but they are really effective when they get to be precisely doing their killing with, let's say, a cancer cells, because your natural killer cells is actually a killer T cell, a type of killer T cell. The cytotoxic T cells are natural killer cells and they recognize other signs of abnormality. uh, Cells that lack the MHC1 proteins, antibodies that are coating target cells. They also have different surface markers that are seen on stressed cells, such as interferon, are actually markers. The NK cells use the same key mechanism as your killer T cells. They actually open the pores, they create a pore, and then they throw in the enzyme or a grenade and makes the cell explode. And they also do what you call an immune surveillance. Your NK and killer T cells are actually prowling around the body looking for markers that they can recognize to be foreign. Now we have something that is not really very popular, the regulatory T cell. This regulatory T cell puts an end to the war zone. If the war is over and we win already, there is no need for this for these soldiers to be around. They need to go back into the lymphatic system, the lymph node, and rest and become memory cells. They don't need to go around because they're going to be using the body's energy, and they might just be killing cells that they're not supposed to be killing, because when they are there and they are soldiers, they are really soldiers. And sometimes they don't know what they're doing if they are actually out there and killing nonsensibly. So the regulatory cells are is a type of helper T cells, and there are different types of helper T cells, and one of them is your regulatory. They are they stop the immune response by direct contact or by secreting inhibitory cytokines such as the interleukin 10 and the TGF beta, which is actually a cytokine that is inhibitory to the actions of the T cells and the B cells. They are important in preventing autoimmune reactions. They suppress the self-reactive lymphocytes in the outside of the lymphoid organ. So when they are not in the lymphoid organ, they need to go back into the lymph node. They need to go back to the hub. They cannot anymore scatter around. If your white blood cell count is elevated because you just had viral infection, it needs to go down to normal. And the effector cells, the regulatory cells, are actually the one that would keep the effector cells away from the plasma and deliver them back move them back into the lymphoid organs, the lymphatic tissues. Here's another table for you to use for your reviews, guys. I could see this, okay, and you need to read on the organ transplant and immunodeficiencies. Examples of this will be your AIDS. I am not going to be discussing this anymore, but they are in your textbook. Read them because I will include them in your test. That's it for this particular part, and we are now finished with our chapter on the immune response.